स्टार्ट करने जा रहे हैं क्लास, ठीक है? Okay. So in the last class, we were studying about, we started with cell to cell communication. Okay. So just a minute. Yeah. Mm. S- So in our last class, we started with cell to cell communication, and in this week, we will be uh, dealing with questions which are related to the further parts of cell to cell communication only. So before everybody joins, we'll just wait so that like one or two people, whoever wants to join, they can join, and then only we'll start the questions. Before that, we'll just see that whenever we talk about cell to cell communication, it's not just about how the cells are communicating with each other, but it is also of how that information, which is once received by the cells, is passed through uh, and it is sent to the nucleus. So, whenever we talk about cell to cell communication, I'm repeating it. Cell to cell communication means how two cells are passing information from one cell to another. And this information is needed throughout the developmental process for the creation of new specific cells, for differentiation, for some kinds of epithelial to mesenchymal transitions, and everything. So these cell to cell communications in a way ensures that the information is passed and indeed it is received by the nucleus. So there are various pathways which are involved in this cell to cell communication, which makes sure that whatever information in the form of a ligand which is received on the receptor, it goes through the cell and it reaches the nucleus so that transcription can take place. Is it fine? So one such pathway, so there are many pathways, there are generally 3 to 4 pathways which are involved in this process. So one such pathway that we have already dealt last week was the RTK pathway, which is receptor tyrosine kinase pathway. So last week also I told this, I'll just be very briefly, I'll be repeating it, that whenever we are discussing about any pathway, uh, the basic things that we need to follow are that there is always a ligand, okay? There is always a ligand, like this is the ligand. The ligand will bind to its receptor. So there is a ligand and the ligand will bind to its receptor. So these are the receptors. So this is the ligand. Ligand will bind to the receptor, and then the cytoplasmic domains of the receptors, they might get activated. So if this is the receptor, this whole is the receptor, this is the cytoplasmic domain, and this is the extracellular domain. Okay. So when a ligand binds to a receptor, the cytoplasmic domain of the receptor, it gets phosphorylated. And when the cytoplasmic domain of the receptor gets phosphorylated, it phosphorylates some other downstream molecules, and a chain of phosphorylation or activation happens. A cascade of events takes place, which ultimately leads to the transcription of the respective gene. This is the active transcription factor, it will transcribe. So it is a general mechanism through which any receptor signaling, any signaling uh, works. Okay, just a minute. Grace, I just wanted to ask that you have raised hand. So is it because you were telling me whether my voice is coming or not, or you have a question? If you have a question, you can ask, or if you don't have a question, you can lower your hand. If you have a question, you can ask, otherwise, you can lower your hand. Okay, fine. So this is what I was telling you, that this is the RTK pathway. Okay, so this we have already discussed last time, that always remember that whenever we talk about this RTK pathway, RAS is the downstream molecule, RAS, RAF, these are the downstream molecules, and further downstream molecule is the ERK. Why I'm telling you? Because there might be questions related to it. Okay, so now we'll move to the questions, and then with the questions we'll try to solve the understand the concepts. Okay, very good evening, Ajay. Very good evening, everybody. So now we'll be starting the questions. So as I was discussing that last week we started with cell communication and we studied that there are certain pathways which are involved. Yeah, I am recovering. I'm better now. Thank you for asking. So yeah, we were discussing some of the pathways, and as I told you that a general principle of how a pathway works, that there is a ligand binds to the receptor, and then there is a cytoplasmic domain and a extracellular domain. The cytoplasmic domain of the receptor, it gets activated, and whenever we are talking about activation in terms of pathways, it is phosphorylation. Phosphorylation is a kind of activation which activates the signaling molecules, which activates the tyrosine residues, which leads to further downstream signaling. So, whenever a ligand binds to the receptor, the cytoplasmic domain of the receptor, it gets phosphorylated, which further activates several downstream molecules, and there is a cascade of events that takes place, which ultimately leads to the transcription of the gene. So this is essentially a general way by which most of the pathways works. The ligand can be different, the downstream
Madam, no
molecule
voice.
can be different, and the ultimate
Hello?
transcription of the gene would be
No
different.
voice.
Hello? Yes? uh, am I audible? Okay, I think so. Grace can hear me. Can other hear me? Just Rina, can you hear me? Okay, I think then there is a problem from Ajay's side, because if others can hear me, then I'll just write it in the message. Okay, fine. Where I was. So this just breaks the flow. Yeah. So so this is the general mechanism of which of how a pathway works. Okay, now we'll come to questions now. So our first question is, premature activation of FGFR3 kinase leads to A. Increased proliferation of hair follicle cells. B. Thanatophoric dysplasia. C. Increased chondrocyte proliferation. Or D. Enlargement of the lips. So think, you have 2 minutes to answer the question. After 2 minutes, I will switch to the screen. Just a minute. I think someone wants to join, or there is any issue. I don't know. Okay. Okay, so I think people have answered it. So just Reena has answered B and C. Ajay has answered B and Benitha has answered B and C. So Grace and Ajay have raised their hands. So is there any query? You can ask if there is any query, or you just wanted to give the answers. I think Ajay, so Ajay is saying that, so Grace, can you hear me? Can others can others hear me? Because Ajay is constantly saying that there is no voice. I think there is some problem from his end. So I'll just. Anyways, I hope that others can hear me. Okay, then I think there is some problem from his end. Fine. So B and C mostly you have answered. That is thanatrophic dysplasia and increased chondrocyte proliferation. So the correct answer is thanatrophic dysplasia. So can anybody tell me like who has answered C also? Uh, yes. So students who have answered C, can they tell me why they have opted for C options also? Can you tell me? Otherwise, I'm going to discuss it. But can you tell me the reason why you have opted for C? C says increased chondrocyte proliferation. So, do you think there will be increased chondrocyte proliferation? See, what happens here is that just look. So, this is the FGF receptor 3. Okay. So, basically, it comes under the JAK-STAT pathway that if there is any kind of mutation in the receptor or the kinase domain, so what happens? Your STAT1 is phosphorylated, and because of this phosphorylation, your growth stops before birth only your growth stops, which leads to narrow chest and extremely short limbs, which can lead to the disease, which is condition, which is thanatrophic dysplasia. So basically, there is a suppression of chondrocyte development and not increased chondrocyte development. Okay, the proliferation of chondrocyte is suppressed because of this mutation. So you can see that thanatrophic dysplasia, it is a rare genetic disease condition that affects a fetus bones and lungs. It is characterized by short stature, underdeveloped lungs, and other skeletal abnormalities. This is your thanatophoric dysplasia, and this is characterized by short limbs, narrow chest, short ribs, and an enlarged head with a large forehead and a prominent eye. So these are the characteristics of thanatrophic dysplasia. Okay, so now we'll move to the next question. So you should know the mechanism also. The question was that premature activation of FGR kinase will leads to. So basically, you should understand that all in developmental biology, whichever pathway is required for development or proliferation, the activation of that pathway should also be time and cell specific. What is the meaning of time and cell specificity of activation of any pathway? It means that premature activation of a pathway can also lead to conditions, diseased condition or some abnormal conditions. And overexpression or overactivation or delayed activation of a pathway can also lead to some conditions. So you need to understand that whichever pathway we are dealing with, the pathway should be activated at a specific time, at the required time. Here, what is happening in this case, the JAK-STAT pathway, which has this FGFR3 kinase domain. This kinase domain is getting activated prematurely, and because of this, it's a JAK-STAT pathway. So if your this domain is activated, then STAT would be phosphorylated, and then because it is prematurely activated, therefore the growth of the cartilage, it stops before birth, which can lead to the condition of thanatophoric trophoric dysplasia. Okay. Next question is, hedgehog binds to dash, which inhibits dash in the absence of the ligand. So your options are A, slimmed and patched respectively, B, patched and pKa respectively, C, patch and smoothened, and D is smoothened and patch. Okay. So you have 2 minutes to answer this question. Okay. Shall I switch the screen? Okay, I'm switching it now. Let's see what you have answered. Okay. So it seems that everybody has answered. It's very good. Yes. So Ajay, Benitha, Jasrina, Grace, and Camellia. Everybody has answered C. What is the C option? Which is hedgehog binds to patch and it inhibits, which inhibits smoothen in the absence of the ligand. It is, sorry. Yeah, that is the correct answer. Very good. So let us quickly see the hedgehog pathway. So you see, so this pathway can be understood in two ways. The first is the when the ligand is absent, and second when your ligand is present. So what is the ligand here? The ligand is hedgehog. So when your ligand is absent, what happens is that this is the patch protein, which is a receptor for your hedgehog ligand. So this patch will inhibit the smoothened protein. Okay. So what I'm trying to say is that patch will inhibit smoothened protein. And because of this inhibition, the CI protein acts as a repressor. The CI protein, it acts as a repressor. Okay. And there is no transcription of hedgehog responsive genes. So this CI protein, it can act as a repressor also. It can act as a activator also. It depends on the presence and absence of hedgehog. When hedgehog is present, that means the ligand is present. When the ligand is present, then CI act as a activator. Whereas if the ligand is absent, then CI act as a repressor. What does it mean? It means that the function of this CI protein is dependent on the presence or absence of the ligand, which is your hedgehog. Here you can see condition B, when hedgehog is present, then what happens? Then smoothened protein is activated. Okay. because when hedgehog is present, you can see that patch protein, it cannot inhibit the smoothened protein now. See, this is your patch protein, and this area, it is completely binding to the smoothened protein, inhibiting it. But as soon as hedgehog comes and binds to it, you see the turn, it is a turn, which means now the patch protein is not completely binding with the smoothen protein, and hence now smoothen is not inhibited. Now once the smoothen is activated, then smoothen will inhibit your pka and slim, and because of this inhibition, the ci protein is not broken into, and because it is not broken, therefore it is a complete whole protein, and now as a complete whole protein, it can activate the transcription of hedgehog responsive genes. Here what happens, that because smoothen is inhibited, so this ci and pk along with other proteins, it phosphorylate the ci repressor, I mean ci protein, and therefore the ci protein is broken down into pieces, and this broken piece, it acts as a repressor. So CI will act as a repressor when it is phosphorylated and broken. Whereas when CI is present as a whole, it will act as an activator. Fine. So this is a hedgehog pathway. Hedgehog pathway again. So like we studied about the RTK pathway, receptor tyrosine kinase pathway, and then is your hedgehog pathway. So up till now we have studied about two pathways which are important in your development, in developmental biology. Next question is, cyclopamine inhibits dash by inhibiting dash biosynthesis. Your options are A. Wnt signaling and beta catenin, B. Hedgehog signaling and cholesterol respectively, C. RAS MAPK signaling and GTP respectively, and D. JAK-STAT and prolactin respectively. So you have to answer that cyclopamine, it inhibits what? By inhibiting what? Okay, you have 2 minutes. 1 minute more, and then I will switch the screen. Okay, now so I'm switching the screen. Hmm. Hmm. So what you all have answered? Let's see. Hmm. So Ajay has answered B, Jisrina has answered B, Grace has answered B, Camelia has answered B, and Benitha. So everybody has answered B. And what is the B option? Yes, so B option is the correct. That is cyclopamine. It inhibits the hedgehog signaling by inhibiting the cholesterol biosynthesis. So basically, cyclopamine is a teratogenic steroidal alkaloid. Now, what is the meaning of teratogenic? Can anybody tell me the meaning of teratogenic? Anybody who knows the meaning of teratogen or teratogenic? Causing cancer. What are teratogens? It causes deformations. Hmm. So basically, the meaning of teratogens is that it causes some kinds of malformations or deformations. So whatever is the structure is not synthesized properly if because of teratogenic agents. Okay. So in very simple word, if I want to explain you, basically it causes the abnormality in the physiology. Okay, there is abnormality in the physiology. For example, you can see here, see, you see such a malformed face. Why? Why this malformation has happened? Because of teratogens such as gervine and cyclopamine, which are found in some plants. They inhibit the cholesterol biosynthesis, and since so that cholesterol biosynthesis has some role in the proper physiological morphology, and therefore the morphology of the face is completely damaged. So teratogens are basically your steroidal alkaloids, which causes some kinds of malformations or deformations in the normal physiological features. And also you should understand that these teratogens, they basically inhibit your hedgehog pathway. Now how are they going to inhibit the hedgehog pathway? So they target the smoothened receptor. Okay, so these teratogens, they target your smoothened receptor. So if you see here the pathway, yes. So what are they doing? That they are targeting the smoothen receptor. This is targeted. So what happens? That even if the hedgehog is present, this smoothen is targeted, and therefore the hedgehog signaling does not takes place. Understand? So even in the presence of hedgehog, because these teratogens are targeting your smoothened receptor, so even in the presence of hedgehog, your signaling is not taking place. So that is what these cyclopamine and other teratogens they do. So the correct answer would be cyclopamine. They inhibit the hedgehog signaling by inhibiting the cholesterol biosynthesis. Okay. Now next question is, calcium binding domain of integrin is present in A. Intracellular domain of alpha subunit, B. Intracellular domain of beta subunit, C. Extracellular domain of alpha subunit, and D. Extracellular domain of beta subunit. 2 minutes. It's a very easy question, straightforward question. Hmm. 2 minutes. Hmm. 1 minute more. Okay, now I'm switching the screen. Hmm. So, okay, so Benita had answered yes. So, teratogens, they causes developmental abnormalities. Correct. So, Ajay has answered C. Benita has answered C. Camellia has answered C. And Jasrina has also answered C. Okay. So, let us see what is the correct answer. C is the extracellular domain of alpha subunit. Yes, it is the correct answer. So, all those who have answered C, it is correct. Now, let us see in this figure. Yeah, you see, the question was calcium binding domain of integrin. So you see, this is your integrin. Hmm. This is the calcium binding sites. Okay. And calcium binding sites, and this is alpha subunit of integrin. And this is the beta subunit of integrin. Can you see here? This is the beta subunit of integrin and this is the alpha subunit of integrin. So this whole is your alpha subunit of integrin only. So where is your calcium binding site present? Alpha subunit or beta subunit? It is present in the alpha subunit of integrin. Now this figure is important because our next question is also going to be from this figure. Fine. So just look at this figure carefully. Then I will switch to the next question. Okay. Now the next question is alpha actinin connects A. Tailin molecules, B. Vinculin molecules, C. Integrin molecules, or D. Actin microfilaments. Okay. So if you have seen that figure that I have just shared, you will be able to answer this question. Okay. So after 1 minute, I will switch the screen. Think. 1 minute more. Okay, now I'm switching the screen. Okay. So what you have answered? Okay. Um, from where is it starting? Okay, let me consider it from 632. So Camellia has answered D, Ajay has also answered D, and Benita has answered D, and Jasrina has answered B and D both. Okay. So what is your the D option, which is actin microfilament, which is correct. So you can see in this figure, again we can just look at this figure. This is the actin microfilament and this is your alpha actinin. So alpha actinin is connecting what? The actin microfilament. This is also actin microfilament. So our question was alpha actinin connects. So you can see from here that this is your alpha actinin and this is your actin microfilament. This is, and from the name itself you can guess alpha actinin. Okay. Now comes a good question. It's a very interesting question. I will give you 3 minutes for you to think about and to answer. Okay, so the question says, which of the following mutations in Wnt will most likely affect its ability to function as a signaling molecule? Okay, Your first option is UGU to UGC, second UGU to UGG, third UGU to UGA, fourth UGC to UGG, fifth UGC to UGA, and last one is UGC to UCG. Okay, so this is a question. I will give you 3 minutes to solve this question. Think and then answer. Hmm. Think. If you have studied in molecular biology about the genetic code, then it would be easier for you to solve this question if you remember it. 1 minute more. After one more minute, I will switch the screen, okay? You just have to know which which codon specifies what. If you know which codon specifies what, then it is very easy for you to solve this question. Hmm. Chalo, I'll be switching the screen now. Let's see whether you were able to answer this question or not. Hmm. So, Ajay. So, Ajay has answered. Apart from A, all options are correct. Okay. Camellia, I think B and D or C and C, D. Okay. So, I will say that Camellia has opted for C and D. Okay, then Benitha has opted C and E. Fine. Jasrina, Jasrina, can you give any answer? What you think should be the answer? This is the question. What you think should be the way to solve this question? Can you think? No? Okay, fine. See, so for this question, there are two concepts that you need to know. First is what kind of mutation uh, gives rise to what, and second is the codon, genetic codon table. Okay. Now, mutations can be of several types. Okay. If I want to study about mutations, they are missense mutations, nonsense mutations, and some are silent mutations. Okay, so mutations can be of several types. These are not just the only types of mutations. There are other categories of mutations also that I'm not dealing. Okay, some mutations are point mutations, some mutations are frame shift mutations also. So there are many different ways in which you can categorize different kinds of mutations. Okay, but now we are going to majorly focus on these three kinds of mutation: missense, nonsense, and silent mutations. So first we'll start with silent mutation. So suppose if a triplet codon ABC, ठीक है, it codes for an amino acid X. Now, if you know a little bit about the genetic code and genetic codon, you must be knowing that one amino acid can be coded by more than one codon. Okay? One amino acid, remember what I'm trying to say, that one amino acid can be coded by more than one codon. So if your amino acid X is there, it can be coded by one set of codon. It can also be coded by another set of codon. So one amino acid can be coded by more than one codon. So silent mutation, suppose if there is a A, B, C. This is your one codon which codes for the amino acid X. And there is change at C, the third base, and now it is, let's say, ABD for example. But this ABD also codes for the same amino acid X. So what has happened? There has been a mutation. C has changed to D. There has been a mutation. But since both the codons they code for the same amino acid, so there is no change in the phenotype. Because earlier, before the mutation, also the phenotype was X protein. After the mutation, also your phenotype is X protein only. That means such kind of mutations are known as silent mutations. That means mutations which has no effect on the phenotype. Is it clear to everybody? What is the meaning of silent mutation? I hope silent mutations are clear to everybody. That means your mutations has happened in such a way that the mutated amino acid also, sorry, mutated codon is also coding for the same amino acid. So it is a silent mutation. Then I come to your missense mutation. The general kind of mutation, missense mutation. So if a codon ABC gets changed to ABD and this ABC codes for X protein, this ABD is coding for Y protein, that means the complete sense has been changed. Earlier the protein was X, now the protein is Y. So this kinds of mutations are known as missense mutations. Then we come to nonsense mutations. Nonsense mutation. So missense means the sense has been changed. Earlier it was coding for X protein, now it is coding for Y protein. Nonsense mutation means that now it is not coding for any protein at all. Okay, now when it can happen? It can happen when your codon is changed to a stop codon. Suppose if there is UAG, UGA, and UAA. These are your stop codons. Suppose there was an amino acid which was UGG. Now, if your UGG is changed to UGA, so it has changed to a stop codon. So, when an amino acid is changed to a stop codon, then there is going to be no sense made. So that kind of a mutation is known as a nonsense mutation. So is this clear? Missense, silent, and nonsense mutation. Can you now differentiate between these three mutations? Yes or no? Just raise your hands if you could understand, or if there's any problem, let me know. Okay, good. So now these three kinds of mutations we understood: missense, nonsense, and silent mutations. Now we'll try to apply this concept in this question. Okay, so this is your question. I will just keep it here and I will keep the question here so that you can understand it. This part, only this part. Now, okay, your question says, which of the following mutations in Wnt will most likely affect its ability to function as a signaling molecule? That means, which mutation will inhibit its effect? Its effect has to be inhibited. So now we'll see. This is the codon table. This is your codon table. So you can see UCAG, UCAG, and UCAG. So how you have to read? This is the first letter. Here it is the first letter. This is the second letter. And here comes the third letter. So if you have first letter U, U, and here U. So this is your U. Then U, U, C, then U A, then G, then C U, C, A, and G. Could you follow how to read this codon table? You should know how to read this codon table. Now you see UGU. Where you can find UGU? This is your UGU. So we get to know that UGU codes for cysteine. And UGC, it also codes for cysteine. UGC, it also codes for cysteine. So which kind of mutation is this? Tell me, from UGU to UGC, which kind of mutation is this? Codon is provided in exam. I don't think that the codon is provided. You have to remember basic. See, here we are talking about Wnt pathway. So in Wnt pathway, your cysteine residues play a very important role. If you have studied about Wnt pathway, then you must know that cysteine residue play a very important role in Wnt pathway. Cysteine residue is translationally modified. So at least you should know the codons that codes for cysteine. If you know the codons that codes for cysteine, that much is enough, at least for the Wnt pathway. Okay. So I don't think that the codon table would be provided, but it is expected from you that whichever amino acid plays a very important role in that particular pathway, at least that much you should know. Okay, because if codon table would have been provided, then there would have been a codon table in this question as well. But since there was no codon table in this question, so I don't think that the codon table would be provided to you. So now somebody has said that to use the pointer. So just I will explain how to read this here again. Now you see how to read this particular table. So this is the first letter. This, from here we will read the first letter. From here we will read the second letter. And from here we will read the third letter. So U, U, U. This is U, U, U. U, U, C. U, U, A, and U, U, G. So this become one. Then from here U, C, U. U, C, C, U, C, A, and U, C, G. This became this table. Likewise, let us try to read this. So how we will read it? A, A, U, A, A, C, A, 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 G. So this block means second letter has to be U. This block means second letter has to be C. This block means second letter has to be A. So in this whole block, your second letter is U. In this whole block, your second letter is C. In this whole block, your second letter is A. And in this whole block, your second letter is G. So this is how you read the codon table. Okay. Now you told, so the students have tell कि ये कौन सा mutation है? It is a silent missense mutation. So many students have said that it is a missense mutation. So now you see, this is UGU. Sorry. UGU. UGU is coding for what? UGU codes for cysteine. And UGC, it is also coding for cysteine. So how it can be a missense mutation? Because both the codons are making same sense. So it is a silent mutation. It's not a missense mutation. It's a silent mutation. So this one is a silent mutation. Now you see again UGU. UGU codes for cysteine. We already know this. And it is changed to UGG. UGG codes for, see here, UGG codes for tryptophan. So what is happening here? Here a change of amino acid is happening. From cysteine, it is getting changed to tryptophan. So which kind of mutation is this? Tell me, B1, which kind of mutation is it? It's a missense. Uh, it's a missense mutation. Okay, because one amino acid is changing to another amino acid. So when one amino acid gets changed to another amino acid, that kind of mutation is known as a missense mutation. When one amino acid is changed to a stop codon or stop amino then it is known as a nonsense mutation. Why we need triplet codon? Just two codons are enough. So for this question, I would suggest you to go and read the basics where you will get to know that why we need codon as triplets, why it cannot be 2, why it cannot be 4, why it cannot be 1, why codons are always triplets. It's a very lengthy portion. I cannot cover it here. You go and you read this. These are very basics that you should know. Okay, you go and you yourself should go and read it. It's a very basic. It's the first thing when you study molecular biology. They tell you by making many permutation combinations, they will tell you that which permutation combination will give you triplet codons, and by which permutation combination will give you enough codons. Okay, anyways, so this is a missense mutation. Now you see second one, UGU. UGU code for cysteine, and UGA it codes for stop codon. So which kind of mutation is this? It is a, it's a nonsense mutation. Yes. So Camelia, you have a question you can ask. Camelia, do you have a question? Okay, fine. So it is a nonsense mutation. Okay. Next, UGC. Where is UGC? UGC codes for cysteine. It is changed to UGG. UGG codes for tryptophan. So this kind of mutation is cysteine is changing into tryptophan. So it is a missense mutation. Then UGC, which is cysteine. Cysteine is changed to UGA. So E1. Which kind of mutation is E? Tell us. E is UGC to UGA. Which kind of mutation is this? It's a nonsense mutation. Why? Because UGC codes for cysteine and UGA codes for stop codon. So it is a nonsense mutation. Then UGC is changed to UCG. Now what does UCG codes for? UCG codes for serine. So cysteine is changed to serine. So it is a missense mutation. So only the first one is silent mutation and rest all the mutation are changing, either it is changing to a stop codon or it is changing to a codon which codes for some another amino acid. So except for A option, all other option will affect your Wnt signaling. Nonsense mutation will cause a shorter protein or peptide to be synthesized, and missense mutations will cause a different amino acid to be synthesized. So the answer would be B, C, D, E, and F. Okay, the question was, which of the following mutations in Wnt will most likely affect its ability to function as a signaling molecule? Except for the first one, all other will affect the signaling because some are silent mutations. Sorry, some are missense mutations, some are nonsense mutations. So these all are mutations. So is this question clear to everybody? Okay, fine. So that is why I started this question. Why how I explained you first the different kinds of mutations, and then we went to the uh, codon table. So therefore, two concepts were needed in this question. First concept was understanding the different kinds of mutations, and second concept was understanding that amino acid can be coded by more than one codon and how to read the codon table. Now, in very briefly, I'll just explain you very briefly because we have touched this topic. So I don't want to leave it halfway. That what are point mutations and what are frame shift mutations? Okay. So point mutation means, can you guess by the name how can it differ? Point mutation means mutation at a particular point. Only particular point is getting mutated. Frame shift means mutation in such a way which is causing the complete frame of the codon to change. For example, if you have, let's say, AUG, okay. Now, point mutation means that, let's say, your G is changed to C and now it has become AUC. Mutation at a particular point has taken place. So G has been replaced by C. G has been replaced by C. And so what happens with because of point mutation? Suppose your codon is like AUGGUC. So this is your codon. This is first codon. This is second codon. So what has happened in point mutation? Your G has been now replaced by C. So your new this would become AUC and GUC. Okay. Now what happens in frame shift mutation? Same example I am taking. You have AUG and GUC. Now in frame shift mutation, what happens? There is either insertion or there is deletion of an particular uh, nucleotide or amino acid. Here what happens? Suppose extra A is added, addition. So now because this extra A is added, so the entire frame after this addition would be changed. Why? Now how you will read it? You will read it A use G. Then because this extra A has been added, AGU and then C with other further. So now this GUC, the frame has changed. Now it has become AGU because extra A has been added over here. So now the complete frame has been changed. Here, whatever change was there, that change was confined to that particular codon only in point mutations. But in your frame shift mutation, whatever change happens, that change is translated to all other codons because now the frame has been changed. Is it clear? Is the difference between point mutations and frame shift mutation clear to everybody? In frame shift mutation, there is addition or deletion that takes place, and because of the addition or deletion of the nucleotide, the entire frame changes. Whereas in the case of point mutation, mutation occurs at that particular point, there is the replacement, and because there is replacement, other codons are unaffected. Okay, now we'll come to the next question. Hmm. Which of the following statements about the transit of the Wnt protein from ribosome to the outside of the cell are correct? Wnt proteins are liposylated on cysteine residues. Wnt protein requires HPGCs in the extracellular milli. Wnt proteins are O-glycosylated in the endoplasmic reticulum. Cholesterol addition to Wnt is required for receptor recognition. Wings are not required to pass through the Golgi, and frizzled receptors contain cysteine-rich residues on the N-terminus. So, you have 3 minutes. Understand this question and then try to answer this question. Okay, 3 minutes to solve this question. Hmm. Read all the options very carefully and then try to understand and answer. Okay. Okay, 1 more minute left. Think what can be the options, correct options. Anyways, we are going to discuss, but think first. Okay, now I'm switching the screen. Hmm? One question I left here. Okay, I was thinking that one question I left. This question is okay, fine. So Ajay has answered A, B, and F. Grace has answered B, and Penitha has answered B, E, F, and A. Okay. So, what we'll do is that one by one, we'll try to understand each and every point, and then we'll see whether those points are correct or not. Okay? Yeah. Mm, hmm. So the first point is Wnt proteins are liposylated on cysteine residues. So is it correct? It is a correct statement. Why? Because Wnt proteins undergo post-translation modifications, which includes the addition of lipid moieties on the cysteine residues, and this modification is crucial for their secretion and signaling activities. So we can say, according to me, this option should be correct. So they are liposylated. Wnt proteins required heparin sulfate in their extracellular environment. Yes. So this heparin sulfate proteoglycans, they actually play a very important role in the extracellular matrix because they help in the distribution, they help in the stabilization, and they help in the gradient formation of Wnt proteins. So these heparin sulfate, they help in stabilization, distribution, and gradient formation of Wnt protein. So this is correct. Then Wnt proteins are O-glycosylated in the endoplasmic reticulum. So this statement, it's even if you do not know anything about Wnt proteins, you can tell that this is a wrong statement. Why? Because in endoplasmic reticulum, only N-glycosylation takes place. O-glycosylation takes place in the Golgi apparatus. So this statement is incorrect. Then cholesterol addition to Wnt is required for receptor recognition. Is this a correct statement or not? This is also an incorrect statement. Why it is an incorrect statement? Because cholesterol modification is not required here in Wnt. Here, lipid modification such as palmitoic acid is critical. Not the cholesterol modification. Cholesterol association we have seen in Hedgehog pathway, not in the Wnt pathway. Okay. So lipid modification takes place, but those lipid modifications like palmitoic acid is added. So that kind of modifications takes place. So this statement is incorrect. Winds are not required to pass through the Golgi apparatus. This is also incorrect statement. Why? Because O-glycosylation which has to occur, O-glycosylation it takes place. But the why this was incorrect? Because here they have written that O-glycosylation takes place in the endoplasmic reticulum. No. In general, O-glycosylation takes place in the Golgi apparatus and N-glycosylation takes place in the endoplasmic reticulum. So this statement is false because winds, they are required to pass through the Golgi. Then frizzled receptor contains cysteine rich residues on the N terminus. So this is this statement is also right, and this particular cysteine residue which is present in the N terminus, it helps in the ligand binding. So this helps in your ligand which is your Wnt ka binding. So your correct options are, this is correct, A, B and F. And A, B and F are the correct options. Okay. So I hope this question is clear to everybody. Just a minute. I thought there was some confusion. No. So is this question clear to everybody? I've tried to explain each and every point to you. Okay. Now let's come to the next question. I forgot I have to take this question first. Anyways, Again, a very interesting question. Now, I'm reading this question. Understand carefully. I will give you 3 minutes to solve this question. Okay. The question says cells in culture were treated with two drugs A and B. So there are two drugs A and B. Cells were lysed and subjected to Western blot with primary antibody which is specific to CI, that is cubitus interruptus. Based on the blot, identify the correct statements regarding the hedgehog pathway. Here we are referring with the hedgehog pathway. Obviously, if CI comes, that means we are referring the hedgehog pathway only. Okay. Your options are A. Drug A can be activator of protein kinase A. B. Drug A can be an inhibitor of protein smoothen. C. Drug B can be an inhibitor of patched. D. Cos2 phosphorylation will be absent in drug B treated cells. E. Microtubule depolymerization will reverse the effect on signaling in drug A treated cells. And last one, microtubule depolymerization will reverse the effect on signaling in drug B treated cells. Okay. So understand this question clearly. It's 7:10 right now. I'll give you 3 minutes. After 3 minutes, I will switch the screen. Okay. Think of all the options. Think the figure which is given to you. We have already discussed the hedgehog pathway and try to relate it with it. Okay. See all the options very carefully. 'See the diagram, the lanes A and B, and then try to relate it with what you know, the concept that you have already for the hedgehog pathway. Okay. 1 minute more. 1 minute more, and then I will switch the screen. Okay. Okay, I'm switching it now. Hmm. Okay. So Camellia has answered A, B, C. Um, Ajay has answered A, B, C, E. Camelia has answered A, B, C. Grace has answered A, B. Benitha has answered A, C, E. And Jasrina has answered A, C. Okay. So remember whatever you have answered. Now, before going to this question, let us quickly, very quickly try to revise about this hedgehog pathway. Okay. So, so you can see important things that we need to remember in this pathway is that cubitus interruptus, which is your CI, it's a key transcription factor which is in the hedgehog pathway. Okay, this CI, when it is not degraded, when it is in its complete form, it can lead to the transcription of the genes. Very important statement. I am again repeating it. When CI is not degraded, when it is in its complete form, it leads to the transcription of hedgehog responsive gene. When this CI is degraded with the help of some kinase, protein kinase. So protein kinase helps in the degradation of CI. It functions as a repressor and there is no transcription of hedgehog responsive genes. Smoothen, this is your smoothen. It is activated when hedgehog binds to it. Otherwise, patch, it inhibits the smoothen. Okay. So in the absence of hedgehog, what happens? That CI is processed. See, in the absence of hedgehog, when hedgehog is not present, just a minute. Huh. When hedgehog is not present, this CI is processed into a repressor form by the action of protein kinase A, COS2 and other proteins. And when hedgehog is present, this smoothened is activated, which prevents the cleavage of the CI. It allows it to enter the nucleus. So protein kinase A, it cleaves CI. And CI is the important transcription factor, key transcription factor. Now we'll try to solve this question. Okay. Now that we have just um, understood the points, now we'll try to solve. Let's see. This is the question. First point is drug A can be an activator of protein kinase A. So what is the function of protein kinase A? To degrade CI. And you can see these legs represent CI. Okay, you see here, subjected to Western blot with primary antibody which are specific to CI. So these antibodies have bound to CI. So in the lane A, we can see that the CI has been degraded. In lane B, we can see that CI is intact. So drug A can be an activator of protein kinase A. Yes, because protein kinase A degrades CI and you can see that CI has been degraded. So we can say yes, it can be true. Then drug A can be an inhibitor of smoothen. Can it be an inhibitor of smoothen? Yes, because smoothen helps in preventing the degradation of CI. So if smoothen is inhibited, the degradation of CI would be more. So yes, drug B can be an inhibitor of patch. So, can drug be inhibitor of patch? Yes. Why? Because patch proteins, they inhibit smoothen and therefore CI is degraded. If patch is inhibited, that means smoothen is activated. If smoothen is activated, that means CI will not be degraded. So drug B, it can be an inhibitor of patch. Then COS2 phosphorylation will be absent in drug B treated cells. So COS2 का phosphorylation, see here. When COS2 is phosphorylated, what happens? Your CI is intact. So when COS2 is phosphorylated, then CI is intact. Here it is showing, it says that cos2 phosphorylation will be absent. No, cos2 has to be phosphorylated, because when cos2 is phosphorylated, then only your Ci can be prevented from degradation. So this is wrong. Now comes microtubule depolymerization will reverse the effect on signaling in drug A treated cells, and it will reverse the effect of signaling in drug B treated cells. So, basically microtubule depolymerization means it is a process by which microtubules break down. That means there is removal of the tubulin heterodimers. Okay. Depolymerization means the tubulin dimers are breaking. So this is one thing about microtubule depolarization. How it is related here? So microtubule depolymerization, it can inhibit the transport of smoothen to cilia. So therefore it blocks the pathway even in the presence of hedgehog. That means microtubule, in a very simple language, microtubule depolymerization, it can inhibit smoothen. So if your smoothen is inhibited, what will happen? Ci would be broken down. So even when your hedgehog is present, if there is microtubule depolymerization which is taking place, then the activity of smoothen is stopped. And because the activity of smoothen is stopped, your Ci does not form. Ci is degraded. So applying that concept, here it says microtubule depolymerization will reverse the effect on signaling in drug A treated cell or B treated cell. It will reverse the effect in drug B treated cells, because in drug A already the Ci is it is already degraded, and microtubule depolymerization will ultimately lead to degradation of Ci. But in A, because the Ci is already degraded, therefore, where the effect would be reversed in drug B treated cells. So, according to me, the answer should be A, B, C, and F. It is A, B, C, and A. Okay. Microtubule depolymerization will reverse the effect of signaling in drug A treated cells. Okay. So, here they are saying it is A, B, C, and E. It's not F. So can anybody tell me if I am missing any concept here? A, B, what you have all answered? A, B, C, A, B, C, and E. So, Ajay, can you tell me if I'm missing any concept? Did I say the opposite thing? See here. I don't know whether he can hear me or not. Okay. So the concept is that microtubule will inhibit the transport of smoothen. Therefore, it will block the pathway activation even in the presence of hedgehog ligands. That means even when hedgehog is present, if microtubule polymerization is taking place, so the smoothen will not be transported, and when smoothen is not transported to the cilia, the uh, pathway activation is blocked. Okay, so it should be E or F. What do you think? Others, can anybody tell me? It should be E or F. Then are you following or not? Are you able to follow or not this question? Just let me know. Are you able to follow or not? Up till D. Are these points clear? Up till D. A, B, C, D. Are these points clear to everybody? Okay. So up till D, I think points are clear. E and F are the problematic statements, I feel. Okay. So here they say that microtubule depolymerization will reverse the effect of signaling in drug A treated cells or in drug B treated cells. Okay. So microtubule depolymerization, it inhibits the transport of smoothen to the cilia. So what it does, It blocks the pathway activation even in the presence of hedgehog ligands. So even if hedgehog is present, see, let us see here. Hmm. Even when even if hedgehog is present, pathway activation does not takes place when microtubule depolymerization takes place. Okay, so according to me it should be F. Um, According to me it should be F, because here the pathway has already been inactivated. There is, because CI is degraded, and when CI is degraded, then there is no transcription of hedgehog responsive gene. So even if microtubule depolymerization will only add on this, it cannot reverse the effect of signaling. Could you understand what I'm trying to say? So I think it should be A, B, C, and F. Okay, fine. This question we have already discussed. Hmm. Now we come to another interesting question. It's a big question actually. This question says two mutated cell lines A and B. This is cell line A and this is cell line B were treated with Wnt antagonist. The following response were recorded. Based on this, identify the incorrect statements regarding the Wnt pathway. Cell line A may have a gain of function mutation in disheveled. This is disheveled and beta catenin. The given treatment will induce Wnt-dependent responses in cell line A but not in cell line B. Cell line B may have a gain of function mutation in frizzled and a loss of function mutation in disheveled. In these cells, the frizzled inhibitor will prevent these responses. Cell line A will respond normally to Wnt proteins. A gain of function mutation in disheveled of normal cells will inhibit Wnt responses. Wnt responses in cell will constitutively expressed beta catenin can be prevented by frizzled mutations. So, I am giving you 3 minutes to answer this question, okay? Listen, read the question very carefully, see the diagrams, and then try to solve this question. It's a good question. Hmm. See all the options and see the graph. Two minutes left more. See all the options very carefully. 1 minute more. Okay. Let's see what you have answered. Hmm. Hmm. Nonitha, is this concept elaborated in Gilbert book, ma'am? So, see, basically, it's a applied question. Applied question means that the concept is just you have to know the wind signaling pathway. Okay, or if you know the wind signaling pathway, then questions are framed on the basis of your knowledge of the wind signaling pathway or any concept for that matter. So basically, uh, in applied questions, like these kinds of questions, they also come in your CSR exams. So in applied question, understanding the concept or understanding the topic is one thing, and implementing the topic or implementing the concept is another thing. So that's why you have to solve n number of questions to be able to get a confidence in these kinds of questions. Okay, so it's like you know the concept, now you have to apply it. You know how wind signaling works, you know what are the factors which activates the transcription of the wind responsive genes, what are the factors which represses the transcription of wind responsive genes. Now you have to apply it. Okay. So wind signaling is given in Gilbert. Wind signaling is given anywhere. On Google, you can go and search it. But the thing is, how you are applying the concept, there it makes the difference. Okay. And Grace has asked another question. Final exam will be descriptive or MCQ. So, as far as I know, it should be your multiple choice questions only. Okay. Now, so students have answered different options. Let's see. Ajay has answered B, D, F, G. Grace has answered B and D. And Camelia has answered B. Okay. So now we'll try to understand the question step by step. Before coming to this question, hmm, I just wanted to have a quick overview of the wind signaling pathway. Because if we'll have a quick overview of this pathway, then it's easier for us to solve this question, right? Now, see, so what we are discussing is the canonical form of the wind signaling pathway. So wind signaling pathway can work in canonical forms and non-canonical forms as well. But this question specifically deals with the canonical form of the wind signaling pathway. So you can see this is the receptor which we in short we call it as FRZ. It is the frizzled receptor. And here the pathway in the presence of wind and here in the absence of wind. So in the presence of wind, what happens? This is the ligand. It binds to the receptor frizzled and another is LRP receptor co-receptor. It activates a protein which is known as disheveled proteins. In short, we call as DSV. Now this DSV, it inhibits this particular complex. GSK3 beta and all. This complex is basically your complex which is responsible for the degradation of beta catenin. So this complex is known as beta catenin destruction complex. GSK3 beta, APC, CK1 and axin. This whole complex is known as beta catenin destruction complex. Complex which is responsible for the degradation of beta catenin. So when Wnt signaling, Wnt protein is present, your disabled is activated. And this activation of disabled inhibits the beta catenin destruction complex. So what we can say that disabled inhibits the beta catenin destruction complex. And what are the components of beta catenin destruction complex? GSK3 beta, CKI, axin and APC. These are the components of the beta catenin destruction complex. So beta catenin is not degraded, it goes to the nucleus and it causes the gene transcription. In the absence of Wnt signaling, what happens? This beta catenin destruction complex, it phosphorylates the beta catenin and this phosphorylation marks it for ubiquitination and your beta catenin is degraded by ubiquitin mediated proteolysis. So Wnt, the presence of Wnt will keep beta catenin intact, whereas when Wnt is absent, this beta catenin destruction complex, it will degrade your beta catenin. Okay, is this clear to everybody? Is this pathway what I just explained, is this clear to everybody? Yes or no? Just give me a thumbs up or something. Fine. So now that we have understood the Wnt pathway, the basics of Wnt pathway, now we'll try to apply this concept in solving our question. Okay, let us go. So this is our question. The question says two mutated cell lines. So these are cell line A and cell line B. Both the cell lines are mutated. Okay, were treated with Wnt antagonists. What is the meaning of antagonist? Antagonist means something that opposes or suppresses the Wnt function. Okay. The following responses were recorded. Based on this, identify the incorrect statement. Here you have to identify the incorrect statement, not the correct statements. Now here you see, this is red one is your beta catenin and your blue one is your disabled. So beta catenin and disabled are should go hand in hand. Hand in hand means because disabled only helps in preventing the beta catenin to get destroyed by the beta catenin destruction complex. Now, first point says cell line A may have a gain of function in disabled and beta catenin. You can see here with time the beta catenin cell line may have a gain of function mutation in disabled and beta catenin. Okay, this looks fine to me. It is fine. The given treatment will induce Wnt dependent response in cell line A but not in cell line B. What do you think? It should be correct or not? It is incorrect. Why? Because you see the given treatment will induce Wnt dependent response in cell line A but not in cell line B. It will induce Wnt dependent response Because although the levels of beta disheveled is less, but beta catenin is still there. Beta catenin levels are still there. And the ultimate, um I mean, the last molecule which has to be upregulated, which has to go inside the nucleus is beta catenin. So whatever mutation has taken place, although disheveled levels has increased, but this mutation has led to increase in the beta catenin level. And since beta catenin levels are good enough, it will go inside the nucleus and it will cause the transcription. So this statement is incorrect. The given treatment will induce beta catenin response in cell line A but not in cell line B. In cell line B also it will induce response. So according to me, this statement is incorrect. Now, cell line B may have a gain of function mutation in frizzled and a loss of function mutation in disheveled. Abhi frizzled ka we cannot say because nothing is there. But yes, a loss of function mutation in disheveled we can see because disheveled is very much down regulated. You can see, okay, it looks fine to me. In these cells, frizzled inhibitor will prevent these responses. Frizzled inhibitor will prevent these responses. Yes, it looks fine to me. Cell line A will respond normally to Wnt proteins. Yes. A gain of function mutation in disheveled in normal cells will inhibit Wnt responses. No, because disheveled helps in the activation of beta catenin. So this is wrong. Wnt response in cell will constitutively express beta catenin can be prevented by frizzled mutations. This looks right to me. Let's see what are the incorrect statements. The given treatment will induce Wnt dependent response in cell line A but not cell line B. Correct. In these cells, frizzled inhibitor will prevent these responses. Okay. Now you see, this is also an incorrect statement. In these cells, frizzled inhibitor will prevent these responses. Why? See, ultimately, whatever mutation has taken place, that mutation has led to an increase in beta catenin levels in both the cases. So no matter whether frizzled is mutated or not, your beta catenin levels are up there because of this mutation. So this is also according to the statement. I think this, because of this reason only, this is a wrong statement because frizzled inhibitor will not prevent the response because this mutation has anyways lead to the activation of beta catenin. Then a gain of function mutation in disheveled of normal cells will inhibit response, that we have already said it is wrong. And Wnt response in cells with constitutively expressed beta catenin can be prevented by frizzled mutations. No, because there has been a mutation in beta catenin because whatever mutations that has taken place. So even if your frizzled receptor is mutated, your beta catenin will continue to be transcribed. So the incorrect statements are these, which is B, D, F, and G. Okay. Now tell me if you could understand or if you could follow up with this question or not, or if there is any problem, then also you tell me. Is this question clear to everybody? Yes or no? Hmm? If there is any problem, if you could not follow any of the statement, let me know. I will explain it again. Otherwise, I will move on to the last question. Is it clear? Hmm? Tell me anything, you tell me yes or no. Which point is not clear? If there is any problem, just let me know. Grace, A, B, C, D is clear. Okay. A, B, E, F, and G are not clear. Okay. See. cell line A will respond normally to Wnt proteins. So cell line A has normal levels of disheveled and normal levels of beta catenin. That means they will respond normally to Wnt proteins. That means they are, because see, when your beta catenin is upregulated, that means Wnt is present. That means these cells were able to respond to Wnt protein and accept those Wnt proteins. Then only your disheveled levels has also increased. Forget about beta catenin, but here you can see that disheveled levels has also increased. So the activation of disheveled says that they have accepted the Wnt protein. See, this is your Wnt. So when your cell line A was able to receive or able to take this Wnt protein, then only your disheveled got activated. Otherwise, the disheveled could not have been activated. Beta catenin is mutated, forget it. But because disheveled is also activated in cell line A, this means that because it has taken up the Wnt protein, that is why disheveled is activated. Disheveled is activated. So we can say that cell line A will respond normally to Wnt proteins because disheveled is activated. Then a gain of function mutation in disheveled of normal cells will inhibit Wnt response. So when your Wnt response is inhibited, when your beta catenin is degraded. So gain of function mutation in disheveled means, so the function of disheveled is to prevent beta catenin from degradation. Gain of function means now disheveled is even strongly it can prevent the beta catenin degradation. So gain of function, that means there are two kinds of mutations: gain of function mutation and loss of function mutation. Gain of function mutation means that this mutation has increased its function. Whatever function this particular protein is destined to perform, that function has increased. Loss of function means whatever function a protein is destined to perform, it has lost it. So gain of function mutation in disheveled means that the normal function of disheveled is to prevent beta catenin from degradation. And gain of function mutation means that now it is more strongly preventing beta catenin from degradation. So when beta catenin is not degraded, then Wnt response will go on continuously. But here it is saying that it will inhibit the Wnt response. That means it is incorrect. Okay, because gain of function mutation will lead to normal Wnt response, even more Wnt response, but not it will inhibit the Wnt response. Then Wnt response in cells will constitutively expressed with constitutively expressed beta catenin can be prevented by frizzled mutations. No, because in both the cases you can see that beta catenin is expressed constitutively, even with disheveled or without disheveled. That means there has been some mutation in beta catenin. So now it doesn't matter whether result is mutated or disheveled is mutated, beta catenin will continuously function. So here it is saying that this beta catenin expression can be prevented by frizzled mutation. No, because beta catenin has been mutated in such a way that it will continuously express. Is it fine? Hmm. Frizzled mutations are not mentioned in the question. Yes, frizzled mutations are not mentioned, but some but from the options you can get to know that there has been some mutation in such a way that beta catenin is getting constitutively expressed. Because you see, even when disheveled is also not there, then also beta catenin is there. How is it possible? Unless beta catenin is mutated, when disheveled is not there, beta catenin should also not be there. That means there has been some mutation in beta catenin which is causing its expression continuously. So it means that it doesn't matter whether the upstream molecules are mutated or not. The downstream molecule, the last molecule is present, which is the most important one, because it is beta catenin is only ultimately going to the nucleus now. So if beta catenin is there, it doesn't matter whether frizzled is there or not, disheveled is there or not, because beta catenin is what we require. So this is the thing. It is a negative question. See, it's a I thought little tricky question, because here first they are saying that they are using antagonist. So antagonist should not be there, and again they are saying that it is a you have to select the incorrect options. So then it becomes difficult to select whether you have to see the incorrect one or the correct one. So there it becomes a little confusing. So the question is little tricky. Okay. Now we come to the last question. Topic expression of ectopic. Which of the following statements regarding cell signaling are correct? Ectopic expression of gap increases MAP dependent transcription. Loss of function mutation in RAF will not only have an effect in signaling in cells with a gain of function MAP mutation. Loss of beta catenin will adversely affect ECM mediated signaling. Cell migration is adversely affected upon mutating glycine residues. Signaling in cancer metastasis is roughly similar to the formation of neutral crust cells. Wind proteins are cysteine rich and require cholesterol at their N terminus, while hedgehog proteins have a cysteine rich C terminus. So, you have to select the correct options. So, I'm giving you 2 minutes. Select and give me the answer. Think. Okay, now I'm switching the screen. Hmm. Um, okay. From where is it starting? Ah, so Ajay has answered B, C, D, E, Benitha, D and C, Jasrina E, B, C, Camellia C, D, and Jasrina as D. And why is A, B repeated? Okay, let me see. Ectopic expression of gap increases MAPK dependent transcription and loss. Okay, just very quickly we'll try to do this question. Okay, so the first is we have to see which of the following options are correct. First is ectopic expression of gap increases MAPK dependent transcription. Is it correct or not? It is incorrect. Why it is incorrect? Why is it incorrect? Because ectopic expression of GAP. What is GAP? GAP is a GTPase activating protein. That means it activates GTPase. And so GAP means GTPase activating proteins. So basically, um, small GTPase like RAS, they are inactivated with the help of GAP activity. So therefore, RAS is inactivated, and since RAS is crucial for MAPK pathway, ectopic expression of GAP, it would decrease the MAPK dependent transcription. Why? Because GAP will inhibit RAS, because GAP is what? GAP is GTPase activating protein which activates your GTPase. Therefore small GTPase like therefore small the GTP such as RAS, it would be inactivated. So this is incorrect. Next is loss of function mutation in RAF will not have any effect on signaling in cell with a gain of function MAPK mutation. So yes, why it is correct? Because I have already told you that when your downstream molecules are this thing downstream molecules are upregulated, then it doesn't matter what has happened to the upstream molecules. So RAF is an upstream of MEPK. So MAPK, if MAPK has a mutation, a gain of function mutation, it will be constitutively expressed, it would be constitutively active, bypassing the need of RAF activation. So this is correct statement. Then loss of beta catenin will adversely affect ECM mediated signaling. Yes, because beta catenin has a role in integrin signaling, and it is very much important for interactions with the extracellular matrix. So beta catenin is responsible or is required for interaction with the extracellular matrix. So if beta catenin is lost, it can disrupt these interaction, which can lead to impaired signaling. So this is correct. Then cell migration is adversely affected upon mutating glycine residues in fibronectin. It is also correct. Fibronectin is a key ECM molecule. Okay, it is a key ECM protein which is involved in cell adhesion. It is also involved in migration, and it is also involved in some other processes such as wound healing. So glycine residues are present in fibronectin, and these glycine residues are important for proper folding and function. So if glycine residues are mutated, it can disrupt fibronectin structure, which adversely affects your cell migration. So this is correct. Then signaling in cancer metastasis is roughly similar to the formation of neural crust cells. Is it correct or not? Hmm. Signaling in cancer metastasis is roughly similar to the formation of neural crust cells. Yes, because we have, if you have gone through Sir's lectures, he has mentioned that epithelial to mesenchymal transition, it happens in your neural crust cells also. It happens in your roughly in the metastasis process also. So this epithelial to mesenchymal transition signaling, it is similar to the in neural crust cells and in metastasis. Then Wnt proteins are cysteine rich and required cholesterol at their N terminus, while hedgehog proteins require a cysteine rich C terminus. No. Hedgehog protein require cholesterol, whereas Wnt protein requires cysteine. So this is wrong statement. So is this question clear? That's E option, not A. I don't know what you're saying. Okay. Okay, you were saying that A and okay, okay, whatever it is. So the correct answer is loss of function, that we have said okay. Loss of beta catenin, cell migration and signaling. Yes. So whatever I have ticked, these are the correct options. Okay. So with this, we complete today's this thing questions. So today's questions were a little tricky, and I understand that these questions require, are there some resources such as question banks can we have access to for study? So I would suggest, like, whatever topics that are there in your syllabus, you can try to solve CSIR questions of that particular topic. Why I'm saying so? Because in CSIR paper you have a section C. So in section C you have lengthy questions, these kinds of descriptive big questions only. So you can try to solve CSIR questions of that particular topic only. Like, suppose we studied about developmental biology, various pathways. So you can try to, try to solve questions of pathways of developmental biology CSIR questions. Like this, you can Google, you can try to find, or if you have like 10 years previous question papers, from that also you can, you can see. There is no other way because the questions are tricky. So you have to, practice questions, otherwise it's difficult, because there are very conceptual questions, not very straightforward questions. So knowing the concept is one thing, implementing the concept is another thing. So for understanding how to implement the concept, you have to try. Any link for getting old exam paper for NPTEL? Actually, I don't have any link for any of the old exam papers. I don't have that link. Sorry. The only thing is that you can solve questions related to this online. Google. You can just Google it, and very difficult to clear exam. No, no, it's not difficult. Don't worry. It's not difficult. If you know the concept and if you practice, it would not be difficult. See, if there are 100 questions, might be that 10 questions are very difficult to solve, but if you have a full grasp and knowledge, at least you can solve 70%. So don't get disheartened, don't get demotivated. Just try to solve questions, try to solve more and more questions. Okay, that is the only way to excel here. So with this, we end today's session. Next week we'll again meet at the same time, and we'll solve some other questions from week 7's class. Okay, so bye everybody, have a nice week.